So the last presentation that I was asked to do today is on pharmacogenomics, the future of clinical trials, new product development, and the practice of medicine. Let me be honest here. This is a very small group, and I know it's the end of the day, so I really would like, if possible, to not have this as a lecture, as a monologue. I have a lot of information that I could go through, and I'm happy to do that, but I really would like to get a little discussion going on, kind of like we did at the end of those couple presentations. And also, the one that I'm doing tomorrow morning, yes, again, I'm on the agenda one more time, that is a workshop, and I guarantee that's not going to be a lecture. I've got a couple of case studies that I'm going to give to you as an audience, and I'm going to ask you, how would you bring them onto the market? That's a workshop. Okay, so again, there's a handout for this presentation. If you want it, just contact me. I'd be happy to send it to you. So here's what I want to talk a little bit about as quickly as I can. What's pharmacogenomics? Is it really a new idea? How will it impact the way we do clinical trials? How will it affect the way we develop new products, primarily new drugs? And how will it change the way we practice medicine? And what are we not going to talk about? This is not an overly technical presentation. Instead, I want it to be more of an inspirational discussion about what we could and hopefully should be doing someday. Maybe not in the next 10 minutes or day or two, but someday. Let's start out with a premise, and that is what we're doing today or even talking about doing in the area of pharmacogenomics or personalized medicine, with all due respect, is absolutely boring, because it represents the lowest of the low-hanging fruit compared to what we could and what we should be doing. Now, again, I'll give credit where credit is due. We're making progress, and progress is a good thing, but it's taking a long time. As I said before, if we set the bar higher, we will achieve more. So let's talk a little bit about pharmacogenomics or personalized medicine. First of all, what is it? Here's a short video illustrating what some people think of is pharmacogenomics. And then we'll dig into it a little bit more. Let me just turn my volume up here. Okay, here we go.
Genetics refers to the study of genetic influences on an individual's response to drugs. In pharmacogenetics, the analysis of a specific gene or group of genes may be used to predict responses to a specific drug or class of drugs. Pharmacogenomics refers collectively to all the genes that influence drug responses, and how genome-wide analysis may be used to identify such genes in the search for novel drug targets and/or key determinants of drug reactions. The effects of a specific dose of a specific drug will differ between individual recipients. A drug that is effective in one person may have no discernible therapeutic effect in another, whilst a third might show a partial response. In some, there may be undesirable side effects. There are multiple contributory factors to such variation in drug response, such as gender, age, body mass, diet, the presence of other drugs, or of particular disease states, and exposure to certain chemicals or toxins, such as cigarette smoke. In addition to these, genetic factors also influence drug response.
Okay, so this is the conventional understanding of pharmacogenomics or personalized medicine. If we take it a step further, here's what the NIH thinks of it. If you happen to like what NIH says, I don't really care, but this is just one group. Personalized medicine is an emerging practice of medicine that uses individuals' genetic profiles to guide decisions made in regard to the prevention, diagnosis, and treatment of disease. So I'm not saying there's anything wrong with that. What I am saying, and what I want to demonstrate to you in the few minutes we have this afternoon, is that is a very limited view of this technology. This is just one small slice of what this technology can do. So this is a good definition as a starting point, but please don't take this as a stopping point, because we're going to take it much further than what the conventional view is here. So pharmacogenomics or personalized medicine, first of all, is it really a new idea? Absolutely not. Doctors have been practicing personalized medicine for centuries. So what's the difference between before and today? The difference is that in the past, we've been practicing medicine from a phenotypical perspective. Phenotypes, if you remember your basic biology, are observable traits. Now we're starting more and more to be able to practice medicine from a genotypical perspective, to make treatment decisions based on your individual A's, T's, G's, and C's that make up the 3 billion letters in your personal genome. So on one hand, personalized medicine is new in a genomic sense, in a genotypical sense. But on the other hand, it's absolutely not new. Kind of like genetic engineering is not new at all. We've been practicing genetic engineering for thousands of years, or some people would argue since the beginning of life on Earth. The difference is that in the past, we've been limited to genetic engineering from a phenotypical perspective. When two plants cross, when two similar animals cross, when a man and a woman come together to make a baby, that's genetic engineering from a phenotypical perspective. But since the 1970s, with the advent of the biotechnology revolution, we're no longer limited to just similar species. Because now we can take a gene, for example, from a fish and put it into a tomato. Why might you want to do that? Well, imagine you're a tomato farmer. One of the things you're really worried about is if there's an early frost, because if there's an early frost, you could be wiped out and you can be financially devastated. So how do you mitigate the risk of frost damage? One way is by putting some heaters in your field to prevent the tomatoes from freezing. Okay, that's one approach. But another approach would be to take a look around the biological universe and ask, how has nature solved the same problem? And if you do that, you might find that deep in the ocean, where it's always really cold, lives a little fish. It's called the Arctic char. One of the things that this fish has learned to do over the millennia, this is classic Darwinian evolution, is to produce an antifreeze-like protein. Well, if there's an antifreeze-like protein in this fish, what must there also be in this fish? An antifreeze protein gene. So we take that gene from the fish and we put it into the tomato, and we end up with a frost-resistant tomato. A company called Calgene in California first did this in the 1980s and was the first company to get a genetically modified food through, I won't say approved, through FDA in the scenario that I just described. As you can imagine, getting a fish and a tomato to mate in a phenotypical sense is a pretty hard thing to do. But getting a fish and a tomato to mate in a genotypical sense, it's actually quite easy to do. All right, continuing on. So how can pharmacogenomics influence or change the way we practice medicine? Let's start out with a premise. With all due respect to my many physician friends, the way we practice medicine today is so primitive it's probably laughable compared to the way it could or it should be someday. This is not nearly as far off as some people think. We are rapidly approaching the point where we're going to be able to use the patient's A's, T's, G's, and C's to make treatment decisions, and I'll share with you one or two examples of that in a moment. But before we do, let's start out with a question. Since this is a regulatory audience, what is the average efficacy rate for drugs across the board? But first, since this is a regulatory audience, what is efficacy? Who can tell us? Let's try to make this interactive. What does efficacy mean? This is a term that you all probably use almost every day. What? Okay. Anybody else want to try out what does efficacy mean? So a lot of people think that efficacy means, does your drug work? That is what efficacy means in the clinical vernacular. That is what I would tell my medical students. But in the regulatory vernacular, that is not what efficacy means. Efficacy does not mean, does your drug work? That's way too simple of an explanation. Efficacy means, does your drug do what you plan to put on the label if it's an existing drug? If it's a new drug. Or does your drug do what is already on the label if it's an existing drug? I'll say this one more time. Efficacy means, does your drug do what you plan to put on the label if it's a new drug? Or does your drug do what is already on the label if it's an existing drug? Efficacy does not mean, does your drug work? So with that understanding of efficacy, what is the average efficacy rate for drugs across the board? And here are your choices. Less than 10%, 20 to 40%, a little over half, about 50%, or virtually all the time, greater than 95%. How many people think that the average efficacy rate for drugs across the board is very low, less than 10%? How many people think 20 to 40%? How many people think 55, maybe a little more than half percent? How many people think virtually all the time, 95%? Some optimists in the crowd. Not to burst some of your bubbles, but the correct answer is 20 to 40%. Average efficacy rates for drugs across the board, 20 to 40%. That is not particularly high. That is also not, in my opinion, something that our industry should be particularly proud of either. for a whole bunch of reasons, not the least of which is the drugs that you take were never designed for you. They were never designed for you, and we'll show you an example of that in a second, please. Uh-huh. No, well, that's a good question. This is an FDA statistic. So whether the FDA looked outside of the United States or not, I don't know. But that's a good question. I'd have to check the reference and see. Correct, the label claims. Why would you assume that any medical product, including drugs, is 100% effective? You know, when you say that you have to prove your label claims, which is true, prove to what degree? Do you have to show that it works in 100% of people? If we did that, we probably would never have a new drug on the market, right? So this is another thing that a lot of people do. They like to not so much on the efficacy side, but on the safety side. People love to define safety in part risk versus benefit. Oh, if the world were so simple. Here's one of my favorite examples. Anybody familiar with a drug called Lamisil? The indication for Lamisil was for foot fungus. As far as I know, not many people have died from foot fungus. And yet a small but not insignificant number of people who have died from the treatment for foot fungus, i.e. Lamisil, because it's incredibly hepatotoxic. It does nasty things to your liver, right? So if safety is simply a matter of risk versus benefit, how do we explain Lamisil? Great comment. Thank you for sharing that. But I'm just trying to demonstrate that maybe the world isn't quite as simple as what a lot of regulatory folks like to think. Okay, continuing on. Consider this. Most treatments are ineffective in up to half the people that take them. In other words, we swallow a pill and then we flip a coin. Putting that in perspective, if your car only started half the time, if airplanes fell out of the sky half the time, I think you know where I'm going with this. There has got to be a better way. Yeah, go ahead. Please, bring it on. I want to have a discussion. Patient compliance is a huge issue, and by the way, not just are they following the directions, but whose directions are they following? Are they following the label, or are they following what the doctor told them to do? And let's not be so naive as to think that the doctor tells them to follow the label. That could never happen in the real world. Again, you appreciate my not-so-subtle use of humor. But kudos to you, because you're starting to realize, you're starting to point out some of the differences between what I call the theoretical world of regulatory affairs versus the real world of the practice of medicine. And I don't know about many of my regulatory colleagues, but I live in the real world. And in the real world, labels don't mean very much, if at all. Yep. What if the doctor does the wrong thing? Or, here's another scenario, what if the doctor does the right thing, but the right thing is not listed on the label? Because as we talked about a little bit at lunch, many of my regulatory friends, they assume what we teach in medical school is what's on the product's label. That's not what we teach in med school. We teach in med school the standard of care. If the standard of care happens to agree with what's on your label, then terrific. But if not, great questions. Please feel free to continue to chime in. I really want to make it as interactive as I can. And by the way, sometimes people say to me after attending one of my presentations, "Oh, Mike, I think I know how you feel about a particular topic based on what you said." And I will often say to them, well, maybe you do and maybe you don't, and here's why. Because in many of my presentations, I will purposely say things that I don't necessarily agree with myself in order to provoke people, in order to get them to think. I won't tell you if I've done that today or not. That'll be up to you to decide. So it's all in the spin. Alan Roses, a former VP from GSK, said the vast majority of drugs, more than 90%, only work in 30 to 50% of the people. I wouldn't say that most drugs don't work. Instead, I would say that most drugs work in 30 to 50% of the people. I hope you all appreciate my not-so-subtle use of humor here, because I don't know what Mr. Roses is doing today, but if he's not in politics, he's truly missed his calling, because this is the classic example of spin. And unfortunately, in the medical world, we do it as well. So I mentioned a moment ago the average efficacy rates for drugs across the board, 20 to 40%. Anybody want to speculate as to the average efficacy rate for placebos? You're forgetting, with all due respect, I find it interesting as a physician, you would say 0, because more than anybody else, I would assume. No, it's not a drug, it's a placebo, it's a sugar pill. Well, according to the statistic that I took, average efficacy rate for placebos is 30%. So let's put this in comparison. Average efficacy rates for drugs, 20 to 40%. Average efficacy rates for sugar pills, 30%. I didn't say that. Incidentally, I find it fascinating. Again, thank you for the opportunity to go off on a tiny bit of a tangent. How so many people assume that if you put a drug or a medical device inside of a patient and they get better, the reason why they get better is because of the drug or the device that we put in them. Alternatively, people assume if you put a drug or a device in a patient and they get sick or they die, the reason why they got sick or they died is because of the device or the drug that we used. I hate to say it, but biology ain't that simple. And as I often say, especially in the biotech companies that I work with, whenever we start to think we understand something, especially when it comes to biology or medicine, that's when I get nervous. As a matter of fact, as I say to my medical students and the attendings standing in the back of the room, they kind of chuckle when I say this. Oftentimes patients improve or get better, not because of what we do to them, but in spite of what we do to them. Something to think about. So what happens when you go to the pharmacy? You get sick, you go to the doctor writes a script for 2, 3, 4 different medicines to make you feel better. You take that script, you go to, here in the United States, a CVS or a Walgreens or whatever it is in your particular part of the world. You swallow those pills, you may or may not get better, and if you do get better, is it because of those pills? Maybe, maybe not. What's the problem with that model? As I alluded to a moment ago, those pills were never designed for you. Because the person coming before you is getting exactly the same pills out of the same bottle. The person coming after you is also getting the same pills out of the same bottle. These products were never designed for you. So, as you've seen some of my other presentations, I like to use metaphors that everybody can relate to. So here's one. Think of buying shoes. Now, this is a metaphor that particularly resonates in the females in my audiences, more so than the men, for some of you might guess as to why. But think of buying shoes. You go to a shoe store, right? Imagine that you go to a shoe store that only sells one size shoes. Let's say size 7 shoes. If you wear size 7, that's terrific. That's great for you. But what if you wear size 5 or size 9 or size 2 or size 12? You're out of luck. Going to a pharmacy today is very much like going to a shoe store that only sells one size shoes. On the other end of the spectrum, we have truly the epitome of the pharmacogenomic model, where instead of going to a shoe store that sells a single size shoes, we go to a shoemaker who measures your foot and builds a pair of shoes specifically for you. Nobody else, specifically for you. Of course, there are advantages and disadvantages to both approaches. I don't have time to get into that here. But for right now, the optimal balance, the pivot point, is somewhere in the middle. I hope in the future, eventually, we will get entirely to the shoemaker side of the spectrum, but we're not quite there yet. And by the way, former Commissioner Mark McKellen at FDA a decade ago, one of my former bosses at FDA, said the direction that medicine is moving is actually towards more individualized therapies through an understanding of the genetic and molecular basis of diseases in individual patients. So these are not new ideas. So speaking of FDA, what is FDA's role in personalized medicine? I talked this morning a little bit about personalized medicine of medical devices, i.e. 3D printing. Let's talk about it now, personalized medicine in the context of drugs. Well, which center at FDA do you think is taking the lead? CDER, that's the drug folks. CBER, that's the biologics folks. Or CDRH, that's the medical device folks. Which center do you think is taking the lead in personalized medicine right now? Most people would think CDER. It's actually not. It's actually CDRH, the medical device side of FDA. Why? Because the thing that is the closest that we have the most of on the market right now are something called companion diagnostics, which I'll talk a tiny bit about, but that's the topic of a whole different discussion. And that is why CDRH is taking the lead. Why CDRH? Because pharmacogenomics and medical devices is possible if we think differently. Okay, so for those that are not familiar with this kind of technology, let me share with you just a small portion of a show that PBS did a couple of years ago on the
a revolution in medicine based on
medicine.
your genetic code.
Each of us will have the chance to have our complete genome sequenced for less than $1,000 in the next 4 or 5 years.
Whole genome sequencing saved Alexis's life. Had that happened at birth, we would have 15 years less pain and suffering.
A new era of personalized diagnosis and treatment.
And we can think about the right combinations of drugs based on the genome.
The goal is to use the cancer genome itself to tell us how to defeat cancer.
But are you ready for the knowledge that comes with cracking your genetic code?
So this is just the trailer. PBS did this Cracking Your Genetic Code a couple of years ago. I think it's still probably available on their website. For those that are interested, I would highly encourage watching it. So how about an example? Well, here is one of the early companion diagnostics, something called AmpliChip. Actually, I'm going to skip that. You know what? I'm going to go to another better-known example in just a second. So, showing the relationship between new drugs and companion diagnostics, let me share with you a couple of excerpts from an article from the New York Times a couple of years ago. And this should especially resonate with those of you coming from the drug side of the world. It's often not enough for pharma companies simply to bring a new drug onto the market. That's gotten easy to do. Regulators and especially insurers, reimbursers, are also prodding companies to develop tests, i.e. companion diagnostics, to pinpoint which patients are most likely to benefit from a particular drug, thereby sparing other patients from the needless side effects and expenses. One of the things that's bothered me about the way we practice in medicine for a very long time is that why do we have to wait until a patient swallows a pill in order to find out if that pill is going to work inside them? We've been practicing medicine like this for centuries, but when you think about it, it makes absolutely no sense. I want to know to a very high degree of certainty, perhaps not 100%, but as close to 100% as I can, whether or not that pill is likely to work in them before they swallow it, because if it's not going to work, I'm not going to give it to them. I'm going to give them something else, right? As we talked about before, we should never be content with the status quo. Pfizer, for example, has a drug for lung cancer. It works wonders in 5% of people. Not so great, and in some cases it's even harmful in other people. That's on the market. Pharma companies can spend millions of dollars to develop a drug and then reap billions of dollars a year in sales with high profit margins, as we all know. Diagnostic companies, these are medical device companies now, typically spend a few $1 million to develop tests, and their annual revenues are about the same level. So it's very difficult from a business perspective to convince a company on the medical device side to do this kind of thing. For pharma companies, the risk is that a test can lower the sales of their drugs by restricting them to a fraction of the potential patients. Clinical trials can be, as we'll talk about in a couple of minutes, made much smaller and less costly by restricting them only to patients most likely to benefit from that particular drug. So let me take what is, go ahead, yeah, please, please. Yep, yep. It's definitely related, and I love the way you think, and here's why. Because personalized medicine, remember as I talked about this morning, how do you do a clinical trial when your intended patient population is only one patient? The orphan drug program, let me say it this way. Personalized medicine, whether we're talking about a drug or a medical device, it's sort of the epitome, it's sort of the extreme of the orphan program. It's the orphan program on steroids, if you will, pardon the pun. right? But again, that gives tremendous insight into, as I talked about this morning, how do we bring these revolutionary technologies to market in a regulatory environment that is clearly not designed to do that. We have to be able to work within the system to do it anyway. So let's take what is perhaps one of the best known examples of personalized medicine, and that is Plavix. As some of you probably know, Plavix is one of the most commonly used drugs in the world. It's a type of anticoagulant. It's not an anticoagulant in sort of the heparin or the warfarin or the Coumadin sense of the word, but it's kind of like that. For those that are not familiar with it, let me share with you this commercial for Plavix, which I think is one of the better examples of a DTP ad.
In check. Bill is a formidable man, but he was no match for something smaller than a bead of sweat. It KO'd him so fast he didn't even see it coming. It was a clot. Like Bill, if you've been hospitalized with heart-related chest pain or a certain type of heart attack, what doctors call ACS, chances are you've had a clot. But now Bill's doctor is helping increase Bill's protection against heart attack or stroke by putting Bill on Plavix. Plavix, taken with other heart medicines, helps provide greater protection against heart attack or stroke than other heart medicines alone by helping keep blood platelets from sticking together and forming clots.
If you have a stomach ulcer or other condition that causes bleeding, you should not use Plavix. When taking Plavix alone or with some medicines, including aspirin, the risk of bleeding may increase. To minimize this risk, always talk to your doctor before taking aspirin or other medicines with Plavix, especially if you've had a stroke. Additional rare but serious side effects could occur.
Ask your doctor about Plavix today, because no matter how formidable you are, you're no match for a dangerous clot.
And by the way, if you heard all of those contraindications at the end, it's kind of like reading the contents of a package insert. As I often say, who, after reading the contents of a package insert, would ever swallow what was in the package? Which begs the question, if a package insert is not written for a patient, and clearly it's not, why do we put it in a package? Simple, because somebody tells us we have to. Call me naive, but that's not a good reason to do anything. Back to Plavix, bottom line, Plavix is a very good drug for a small number of people. For a whole bunch of people, it doesn't do anything. And for some people, it's actually dangerous. So once again, why do we have to wait until the patient swallows the pill, in this particular case Plavix, to find out whether or not it works? And there was a warning label several years ago that was put on Plavix. Studies have shown that Plavix, a popular heart medication, causes potentially fatal hemorrhaging, including severe gastrointestinal bleeding. My response to this is very simple. Duh. I mean, do you really have to have an MD after your name to appreciate that this is expected? I mean, this is common sense. You don't have to know anything about regulation to know that this is common sense. And as a result, we start to get these kinds of ads showing up on the TV. Watch this.
Medical studies show that patients who take Plavix are at an increased risk of serious bleeding, including hemorrhagic stroke, TTP blood disorder, gastrointestinal bleeding, bleeding from ears, eyes, and nose, bleeding ulcers, subdural hematoma, and death. The FDA recently issued a new black box warning for Plavix. If you or a loved one suffered serious side effects or died after taking Plavix, you may be entitled to money damages.
You get the idea. Once again, I say, duh, this should be a surprise to nobody that knows anything about biology or physiology, because it's totally predictable. Bottom line, one of the big advantages of personalized medicine, in this particular case, companion diagnostics, is we can mitigate all of the problems that they just talked about by prescribing the drugs that we already have today in a more intelligent fashion. Now, since this is a regulatory audience, here's an interesting regulatory question. Are companion diagnostics combination products? Well, one type of combination product currently listed in the Code of Federal Regulations, it's one of three, is what's called a cross-labeled combination product. A cross-labeled combination product is any drug, device, or biologic packaged separately, but according to its label is used with another individually specified drug, device, or biologic, and both are required to achieve its intended use, indication, or effect. I mentioned in my intro this morning, I work as a consultant for Health Canada as well. Health Canada hired me several years ago to help them develop their official Canadian policy for combination products. We spent the better part of a day in Ottawa several years ago trying to decide how Canada wants to regulate this particular kind of product. That's a topic of a whole different discussion. But bottom line, it's not a simple thing. And the reason why I have the word both in here is because the word both is tremendously problematic. Because one could easily argue that a physician can prescribe Plavix without a corresponding companion diagnostic. We've been doing it for a long time. But why the heck would you order a companion diagnostic if you didn't at least plan on or possibly have the patient take Plavix? So it's the bidirectional nature of this definition that's problematic. So are they regulated this way here in the United States? Well, the short answer is it depends. Sometimes they are, sometimes they're not, and it's really holding us back. Well, that would be a different indication of the same drug, or actually that's not a drug, it's a biologic. Technically now it's not even a biologic, it's a biotech drug. But cross-labeling, at least in the context of combination products, means that you have a medical device pointing to a drug or a biologic and vice versa. So if you were, for example, delivering Avastin with a particular catheter, and you said, use Avastin, deliver Avastin with, Boston Scientific catheter, use Boston Scientific catheter with Avastin, then you certainly could construe a cross-label combination product. Of course, I would advise any manufacturer absolutely not to do that because you're complicating your world from a regulatory perspective. You can avoid that. So back to pharmacogenomics and practice of medicine. I don't want to spend too much time on this because this is not a medical school audience, but how does this impact, or how can this impact what we do in the practice of medicine? So here we have a group of patients. Let's say they all have a particular disease. The disease I like to use to illustrate here, let's say hypertension, high blood pressure. Does anybody know how many drugs that we currently have on the market in the United States that have as part of their label antihypertensive? More than 170. More than 170 drugs that are labeled as antihypertensive. So how does a physician, even a cardiologist, know which drug to give which patient? Although, yes, there are some guidelines, you might as well throw a dart at a dartboard. It's about as scientific as that. So here we have a group of people that all have a common disease, let's say hypertension. The people in the blue and the green on the left, those are the folks that we give a particular drug to. And the people in green in the middle, those are the ones who respond positively to that particular drug. The blue people are either not responding at all, or in some cases they might be responding negatively. So is there a better way that we can do this, a more intelligent way to practice medicine? Well, what if we can genomically or enzymatically slice and dice the population of patients such that we group them into subgroups or cohorts, if you will, that are more similar to one another genomically or enzymatically than to the others? So for example, the yellow people are more similar to the other yellow people than to the brown people. And so we give the yellow people the yellow pill. The brown people, we give them the brown pill. The purple people, we give them the purple pill. Not necessarily Nexium, but you get my idea. So one way we can use this technology is to take the drugs that we already have and use them in a much more intelligent way. But I'm going to share with you what I think is the really cool part at the end, how we can use this technology to develop totally new, totally novel drugs. So how long will it take for personalized medicine to impact the practice of medicine? This was a survey that was done a couple of years ago. Most physicians said it's going to take a decade or more for this technology to impact. Well, but it's not so simple because I view pharmacogenomics as a platform technology. So what's a platform technology? Anybody hear this phrase? What's a platform technology? Okay, a platform technology is a new technology or a new way to look at the world that could potentially be used for many different applications and could change the world in some very huge ways, both in ways that exist today as well as in ways that we haven't even dreamed about yet. Platform technologies are truly game changers because it forces people to think differently, what Stephen Covey said, a paradigm shift. Examples of platform technologies include things like sanitation and electricity and automobiles and computers and the internet. What did we do before Al Gore invented the internet, right? So these are platform technologies. Think about this example. Once upon a time, most people thought it was impossible for people to fly. Fortunately, Orville and Wilbur Wright didn't believe this. They were the first two credited for inventing and building the world's first successful airplane and making the first flight 100 years ago in 1903. The question is, how long did it take for human flight to impact the world? Years, decades, centuries? Has their discovery, has their invention stopped impacting our world today? Absolutely not. If Orville and Wilbur Wright were content with the status quo that existed in their world back in 1903, what would we not have today more than a century later? That is why I put pharmacogenomics in this particular category of a platform technology, because we shouldn't be content. So let's see how this can be used as a platform technology in clinical trials. Again, this is a regulatory audience. You're all very familiar with the way we do clinical trials today and the way we've been doing them for decades. But let's be honest, the way we do clinical trials today really has not changed for a very, very long time. The question is, can we do better? Here's a question. Why do clinical trials, especially drug trials, typically require so many people? Hundreds, thousands, sometimes tens of thousands of people. Merck not long ago finished a clinical trial, one clinical trial, 70,000 patients in a single clinical trial. In my opinion, that is nuts, and that is not the future. So the question is, why do we need so many patients in a clinical trial for a drug? I'll give you a hint. We can answer this question in one word. It starts with a V. Say it again. Close. Close. Variability. Variability, because anybody that's taken any statistics course, I hope, would appreciate that the more variability that you expect in a system, the more data you have to collect in order to control for it, in order to analyze it. But there's a problem with that. If it weren't for the great variability among individuals, medicine might as well be a science, not an art. There's a problem with that approach. If you always did what you always if you always do what you always did, you'll always get what you always got. Or put a slightly different way, if you always think the way you always thought, you will also always get what you always got. So if you continue to think of clinical trials the way we've been doing them in the past, with hundreds or thousands or tens of thousands of patients, that's all we're going to have in the future. But if you back off and you say, gee, is there another way that I can skin this proverbial cat? You might be amazed at what you come up with. So how does this apply here? We should always question the status quo. Can we do a clinical trial, never mind for a Me Too, can we do a clinical trial for a new molecular entity? Sorry, that should say molecular, not medical. Using just a few dozen patients, maybe even just 12 patients? Well, once again, you will never do it if you think of clinical trials the way we've been doing it in the past. But if you open up your mind to some new ideas and new ways of thinking, you might be amazed. So how can we do this? Coming back to variability. Variability can be managed or controlled in two ways. I like to think about this like a train. When we manage variability in a clinical trial after the data has been collected, that's like focusing on the caboose, the back end of the train. But what if instead we focus on the front end of the train, the engine? What if we put pharmacogenomic information, I'm not going to talk about how we can do this, but suffice it to say we can, in the inclusion and exclusion criteria of our clinical trial, so that before we even enroll any patients, we selectively rule in and at the same time selectively rule out patients that are likely or not likely to respond positively to our particular drug. If we do that, now we have a way of doing a clinical trial for a new molecular entity with not hundreds or thousands, but a couple of dozen or less patients. That, coming from Boston, is what I would call wicked cool. So when you think of pharmacogenomics in clinical trials, think of the childhood book, The Little Engine That Could. I think I can, I think I can. FDA has put out now a growing number of guidances. I'm going to go through these very, very quickly, just to show you that this is not nearly as far off as some people think. Unfortunately, what's really holding us back, in my opinion, is that most companies, including big drug companies, they don't want to be the first through the doors at FDA with a new product, or in this case, a new technology, a new way of doing a clinical trial. They would instead rather be the second or the third or the fourth for all the obvious reasons. Me, being the contrarian that I am, I love to be the first through the doors at FDA, because if I'm the second or the third or the fourth and I want to do something different than what the people did before me, now I have to argue that what they did before me was not appropriate or perhaps even wrong, and what I want to do is better. As opposed to if I'm the first, that gives me an opportunity to get FDA to think the way I want them to think. And that is a huge advantage. Don't get me wrong, I've been successful both ways, but I would much prefer to be the first rather than the second or the third. So how else can pharmacogenomics impact clinical trials in some huge ways? I mentioned placebos a couple of minutes ago and the placebo effect. What's the placebo effect? You know, as you all know, it's in the mind, so to speak, or is it? What if I could tell you there was a genetic predisposition to the placebo effect? In other words, if you carried a gene, and most likely this is not monogenic, this is most likely polygenic, if you carried a certain combination of genes, you would be more likely to respond positively to a placebo than somebody that didn't carry that combination of genes. Now again, let me be clear, the jury is still debating on this, but there's a growing amount of evidence to suggest that there's at least a possibility that the placebo effect does have a genetic predisposition. So if it does, who would have even considered such a question? I asked this question a very long time ago, but more importantly, how can or how should we use this information? For example, do you think drug companies would want to rule out patients in clinical trials that are most likely to respond positively to a placebo? Of course they would. Why? It's obvious, because that way you can even further reduce the number of patients that you use in your clinical trial, right? The question is, if we do something in a clinical trial that we're not prepared to do after the trial is over and the product is on the market, what happens to our efficacy? Our efficacy of our product drops like a brick. And there have been a number of products, not just drugs, but medical devices as well, that have gotten terrific efficacy results in a clinical trial, but then they get abysmal results in the real world. Here's one example of that. So if there is, and again, this is a big if there is a genetic predisposition to the placebo effect, is the placebo effect simply all in the mind, as a lot of people would think? Yep. Well, you've used the word pointless, let me be clear, not me. But let me answer your question in a slightly different way. Why do we use placebos in clinical trials? A lot of people think it has something to do with statistics. It has nothing to do with statistics and everything to do with healthcare policy. And here's why. Because the regulation says that in order to bring a new product onto the market, whether it's a device, drug, I don't care what it is, do we have to show that it's better than what we already have? No. The regulation says we have to show that it's better than what? Nothing, i.e. a sugar pill. So think about this. If Congress were to decide to raise the bar, and by the way, I do not advocate this, but if they were to decide to raise the bar, and they could easily do this in a nanosecond, that it's not sufficient anymore to show that your new drug is better than nothing, you must show instead that it's better than what we already have. How many placebos do you think we'll be using? 0. So is the real reason why we use placebos in drug trials, a little different than in medical devices, but especially in drug trials, really to do with statistics? Not so much. Okay, so what I think is the coolest application of this technology is in new product development. And again, let's start out with a premise. With all due respect, and I know some of you do this, the way we develop new drugs today is so primitive it's laughable compared to what it could or what it should be. Some of you may remember the old joke, the first pill off the manufacturing line costs $800 million, or whatever number you want to use. How much does a second pill cost? 30 cents. For decades, the pharma industry, and in a similar way, the medical device industry, has put the value proposition on the pill, on the device. As I said this morning, the more pills, the more devices that we sell, the more money we make. That will never work in personalized medicine. And believe me, if we can't sell personalized medicine from an economic perspective, I don't care about getting it through the FDA. That's the easy part. Selling it to CMS and to the insurers, that's a much different thing. So if we instead put the value proposition on the process, make our money on the process rather than the pill, that's the way to solve this economic problem. We've been focusing on the wrong side of the equation, and it's really holding us back. So where do most drugs come from? We have not designed or invented them. Most drugs that we use today, and biologics as well, they come from nature somewhere, from the rainforest, from the ocean. Here's the problem with that. Discovery is not the same as invention. In other words, if you were an engineer, a car engineer, for example, working for a car company, Mercedes-Benz, and you asked your boss asked you to design the next generation of Mercedes-Benz, would you go out to the rainforest to look for your next generation of car design? Of course not. That doesn't make any sense. To me, going out to the rainforest to look for a new drug makes about as much sense as going to the rainforest to look for a new car design. So instead, what would you do? As an engineer, you would sit down at a computer, you would draw out the shape of the body and the headlights and the engine and so on and so on. Why can't we take exactly that same approach to new drug development? Why can't we literally design a new molecule from the level of individual atoms on up that was designed specifically to do whatever problem that we're wanting to solve? That is not the next evolutionary advance in medicine. That is not going from Prilosec to Nexium. That is not going from one bare metal stent to another. That's a revolutionary advance. That's a whole change in the way the ethos of how we approach medical problems. So why not, and again, I'm not going to talk about how we can do this, but suffice it to say we're starting to take all of these billions and billions of A's, T's, G's, and C's, as Carl Sagan would say, and take this information and put it into some supercomputers. Some of the most powerful computer applications on Earth are not in the military, not in NASA. They are in biotech labs, doing something called in silico biology. In other words, why limit ourselves to doing biology in a living system, i.e. in vivo, or in glass, i.e. in vitro? Why not do biology in a computer? Why not teach the computer to read that sequence of A's, T's, G's, and C's, and then use that information just like a cell would to express a protein? And once we do that, then we can literally design a new drug. Again, I can't emphasize this point enough. A new molecule from the level of individual atoms on up, designed specifically to block, interfere, change, whatever you want, what's happening in silico. That is wicked cool. So the epitome of pharmacogenomics is being able to take a much more targeted approach. In other words, instead of taking a one-size-fits-all approach, we take a personalized approach. This pill has my name on it, Michael. You want a pill for you? Put your name on it, and so on and so on. This sounds Star Trek-y, but we're getting closer all the time. Is personalized medicine possible? Some people have said, aren't we exaggerating the personalized medicine approach? I'll be the first to admit there's a lot of hype. But I also strongly believe that this is definitely achievable. But as I said this morning with some of the other examples, the first thing that has to happen is we have to think differently. So I know we're over. Does anybody want to continue with this? You think this is interesting? I'll try to get to the end of it as quickly as I can, but if you do have to leave, I'll certainly understand. Let's take this even further. So we've got to think about the world in which we live, especially for those of you coming from the drug side of the world. How are drugs made? They're made in large quantities. When are drugs made? They're made long before we need them. And finally, where are drugs made? They're made in far-off manufacturing facilities somewhere. The question is, we've been doing this for a very long time, but does it make sense? Not to me. There has got to be a better way. And this ties in now to something very similar to pharmacogenomics, and that is pharmaceutical compounding. Pharmaceutical compounding is basically, and we've been doing this for hundreds of years, a pharmacist sitting in the back of a pharmacy mixing some pills or some potions specifically for a particular patient. We've been doing this for a very long time, but just 2 years ago in the Boston area, the case of the New England Compounding Center tragedy, they changed all of this. For those that are not familiar with this case, and you should be, it's one of the most important regulatory cases in the last few years.
13,000 people nationwide may have received contaminated
back this up
steroid.
because I want you to hear the beginning.
13,000 people nationwide may have received contaminated steroid injections to relieve their chronic pain. And now stress levels are high.
I've heard of people, frankly, waking up in the middle of the night, kind of concerned about it.
The New England Compounding Pharmacy in Massachusetts manufactured the steroid used for pain management between May and September. It's a critical time frame in which investigators say many patients received injections contaminated with the fungus called Aspergillus.
We are cautiously optimistic that many of the efforts to reach patients and clinicians with information about this outbreak are succeeding.
Aspergillus can lurk silently in the body for up to a month before showing symptoms of a rare but non-contagious form of meningitis. It sickened more than 100 and killed at least 8, like Diana Reed of Nashville, Tennessee.
I lost a treasure of a friend. I lost my best friend.
The outbreak has led to scrutiny of federal regulation of compounding pharmacies. They're run by licensed pharmacists and can sometimes make medicines cheaper than major drug makers. But the Food and Drug Administration has very little regulatory oversight of compounding pharmacies.
They seem to have fallen into a gap, and this is the problem that's resulted.
For now, all many patients can do is worry and wait to see whether they too will develop this rare and deadly meningitis. Erica Edwards.
Now, don't get me wrong. This is a terrible tragedy, no question about it. Thousands of people were infected. Hundreds of people lost their lives. It's a terrible tragedy. But how do we respond? Oftentimes when things like this happen, people say, this is a terrible thing, we should prevent it from happening again, which is certainly an admirable goal. And what they typically want to do is they want to create more regulation to try to prevent it from happening, which once again is an admirable goal. But when you create new regulation to try to prevent a problem like this, what other technologies are you going to make it more difficult, if not impossible, to bring to market in the future? And that's where we're going with this. We've been doing compounding forever, but we have problems with it now, and the root cause of this problem, I would argue, is that in the thousands and thousands of pages of regulation on pharmaceutical manufacturing, not once in there does it define what manufacturing means. Some of us talked about this a little bit at lunch. In a nutshell, if a company is making millions of pills in a factory somewhere, most people think that's manufacturing. On the other hand, if you're only making one pill in the back of a pharmacy, most people would say that's not manufacturing. Okay, so if millions of pills is manufacturing but one is not, that implies that there's a line that you can draw somewhere in between, such that as long as you stay on one side of the line, you're not manufacturing, but once you cross it, you are. In those thousands of pages of manufacturing regulation, not once do we define manufacturing. And by the way, I'm not advocating that we should define manufacturing. I'm just simply pointing to what I think of as an engineer as the root cause of the problem. So sorry, go ahead, please. Yep. Please. Good. Good. No, please. You're thinking. And believe me, 5:15 in the afternoon and you're still thinking. That's a good thing. Mm-hmm. Mm-hmm. Correct. Yeah. Yep. Well, I'm going to take that a step or two further, but in the meantime, I'm sorry, remind me your first name again? Miriam, thank you, Miriam. I should know that by now. Miriam, what you're sort of alluding to is, everybody today talks about and focuses on GMP. There's tons of presentations on, one aspect of it or another. But if the future of medicine is personalized, whether it's for drugs or devices, here's the question for you. What's going to be the role of GMPs in the future? Will we have GMPs? in the future. So if any of you have read my bio, you know that I teach part-time at several different schools, including in several regulatory programs, graduate level. And believe it or not, there are a couple of schools now that offer a PhD in regulatory affairs. And I was asked by one of them, would I teach at the PhD level in their regulatory program? And I said, sure, I'd be happy to talk to you about that as soon as you answer for me one question. What the hell am I supposed to teach? And again, I'm being serious. I don't mean to be overly flippant. What the heck does a PhD in regulatory affairs even mean? Is it somebody that can regurgitate more of the regulation than somebody that only has a master's degree? Gee, I hope not. I hope what a PhD in regulatory affairs would mean is that you're willing to talk about and to think about some of these other questions. Like, yeah, we've had GMPs for a long time, but in the future, does it make sense? Yeah, we have this, what I call silo structure of FDA, for example, where we have a center for devices, a center for drugs, a center for biologics. The question is, does it make sense? Some people have suggested, here's an interesting question, do you want a different group of people regulating statins and stents when at the end of the day, they both accomplish the same thing? So some people have suggested, gee, maybe a center for devices and a center for drugs and a center for biologics and for foods and cosmetics. Now tobacco is the 8th center that was created by FDA. Instead, we should focus on clinical specialties, like, for example, having a center for cardiovascular products, having a center for neurological products, for oncology, for gastroenterology, for ophthalmology. There are advantages and disadvantages. I would like to think that if somebody does a PhD in regulatory affairs, dare I say it, regulatory science, that those are some of the things that they would consider, not memorizing more regulation. I would not say that it's pointless. I would say that there is merit to it with one big caveat, and that is, as I said, they should not be able to regurgitate more regulation than somebody that has a master's degree or somebody that has an RAC after their name. One would like to think, but some people would call me, never you, Miriam, but some people would call me naive. Yeah, please. Failing to give what? I'm sorry? Okay. Of course. Well, it's a great question. Actually, if you come tomorrow to my workshop, I'm going to show you an example of an actual product under development, very, very early stage. Okay, but I'm going to show a product of how we can actually do that in a patient. In this particular case, a cancer indication. It's, if you can imagine a drug-eluting stent. This is a drug-eluting stent taking many steps beyond. So come back tomorrow. We'll talk a bit about that. I find it interesting, by the way, you make a comment, well, we've got enough. I think you even said we've got too many drugs. Well, we've got a whole bunch of basically the same drugs, but we don't have very many new drugs. That's a whole other problem. Yeah, please. So in the case of the New England Compounding Center, the crux of the problem was they were doing personalized medicine. They were doing pharmaceutical compounding on a large scale. Personalized medicine on a large scale. Once again, how do we define manufacturing? If we have one 3D printer sitting in an OR and we make one device for that patient, that's one thing. What if a medical device company has 10,000 3D printers in a great big room and they're all running in parallel and they're all making medical devices? Personalized medicine at a large scale, right? We always got to look for similarities where no similarities seem to exist. There's huge similarities between personalized medicine of 3D printing, for example, and of pharmacogenomics? Great questions. Thank you. Please continue. So should FDA regulate compounding? This is not a simple question, and I made this slide prior to the New England Compounding Center tragedy 2 years ago, but as late as 2010, the U.S. Supreme Court said that the FDA does not have jurisdiction over compounding because they don't engage in manufacturing, whatever that means. I actually agree with the Supreme Court decision, but for a slightly different reason. They actually don't regulate, or they should not regulate compounding because it is the practice of medicine. Now, if we want to, as some people have suggested, if Congress wants to give FDA the authority to regulate certain of these areas, like pharmaceutical compounding, that's fine. But that's not something that the FDA can decide unilaterally. That is a matter of healthcare policy. Bottom line, what defines manufacturing? How many pills, how many widgets do we need to make before it's manufacturing? Now, as I said this morning, I'm a contributing editor to 4 of the largest medical technology and regulatory publications in the world. I published a column 2 summers ago on the New England Compounding Center tragedy when this happened. Here's a quote from that article, and I'll share you why in a moment. It's easy to measure the number of people that are harmed by a particular drug or a medical device because that product is not safe enough, whatever that means. But what is much more difficult to measure is how many people are harmed or killed because they don't have access to a drug or device because we've raised the regulatory burden to the point that companies, perhaps like some of the companies represented in this room, decide it's just not worth it to them to bring this product onto the market. That one quote got me invited, and then subsequently invited a second time to talk about this problem on Capitol Hill. And if you think it's interesting dealing with the FDA, which I spend a lot of my time doing, and dealing with CMS as well, Capitol Hill, that's a totally different story. We're talking about the balance here between regulation and innovation. How much regulation is enough? How much regulation is too much? That is not a simple question. And the ramifications go well beyond the compounding case. Many of the drug companies and even the medical device companies that I work with, when I talk about the compounding center tragedy, they say, well, we're not a compounding company. Why the heck do we need to worry about this? They're going to have to worry about it in some profound ways. So what are the ramifications for the future here? Let me share with you my vision of the future of what I call high-tech compounding. So in the old days, pharmacists used to compound products specifically for a given patient, i.e. they would combine a variety of substances to a tablet or a liquid that was unique for that particular patient. This is the way we've practiced personalized medicine for centuries. Yeah, go ahead. Yep. Yep. Sure. Sure. Yep. And the closest thing that we have to that in the United States is what we call nutraceuticals, which are not pharmaceuticals. Nutraceuticals, things like ginkgo biloba and echinacea and St. John's wort, those are not pharmaceuticals, those are nutraceuticals. But again, in my opinion, we're balancing on the head of a pin, right? Because Hippocrates said, let your food be your medicine and your medicine be your food. Whether you call it a nutraceutical, a food, or a pharmaceutical, whether you call it a cosmetic or a cosmeceutical or whatever it is, we should be focusing on what makes sense rather than what we call these things. That's really holding us back. Anyway, great question. So today, compounding has become almost a lost art. Its regulation is not well defined. It's similar to what an example of the polypill. For those that are not familiar, here's the idea of a polypill. We have a number of different drugs that we sandwich together in the same delivery system. I think, with all due respect to my many friends in the pharmaceutical industry, thinking of a pill as a pill is antiquated thinking. We should think about a pill as a delivery system, just like a drug-eluting stent. It's exactly the same thing. As a matter of fact, never mind a polypill. Let's think about a simple aspirin. Can we argue that a simple aspirin tablet is a combination product? Of course we can. The active pharmaceutical ingredient, the API, is the aspirin, is the drug. Everything else along with it is the carrier. It's what we use to deliver it. So in that sense, and this is using regulatory logic, which is very powerful when you use it properly, we could argue, I'm not sure why you would want to, but we could argue that a simple aspirin is a combination product, just like a drug-eluting stent. So the polypill, in the idea, this is a cardiology indication, I won't show you the video, is the idea we take a bunch of different medicines, we put them into the same form. Is this same delivery system? Is this a combination product? Not in the regulatory sense. It's a combination therapy, but it's not a combination product. But here are the regulatory implications. If you get sick and you go to the doctor and the doctor says you need to take 5 different medicines and you swallow those 5 pills separately, does FDA have anything to do with that? Absolutely nothing, because that's purely the practice of medicine and FDA has nothing to do with that. And yet if we take exactly those same 5 medicines and put them into one delivery system, does FDA have any involvement? You betcha. They're all over it. Is that irony? If that's not irony, I don't know what is. And oh, by the way, not only have we not created incentives for companies to do this, we've actually created disincentives for companies to do this. Why? Because if you manufacture 5 different medicines separately, are you required to test for interactions? No. FDA did put out a guidance about 2 years ago about interactions, but for the most part, you don't have to worry about interactions. But if you take these exact same 5 medicines and put them into one delivery system, are you required to test for interactions? You betcha. So why the heck would any company even want to do that? Back to my high-tech compounding idea. Go ahead. Okay, with computer simulation and so on, but still, even with in silico biology, that's just another way of saying computers, companies still don't want to do that. I mean, let's be honest, why would you if you could avoid it? And I hate to make a regulatory recommendation to a company that holds the technology back simply to make the regulatory path easier. Because again, how does that make the world a better place? That's a problem. But if you make, a pill for one patient, how much are you going to make off of that one pill? Please. Say it again. I'm not saying it's good or bad. Remember, as I said this morning, my philosophy of education is to teach you how to think, not what to think. So. I don't know about that. I'm just simply trying to point out what I think is an irony here, and that is if we manufacture these things separately, the regulatory burden, let's be honest, is much lower than if we take exactly those same things and put them together. Multivitamins. Yep. Yep. Can one not make the same exact argument with a lot of the nutritional supplements when we put 20, 30 or more different things in the same, and we've been doing that for decades. Good for you, you're thinking. You're thinking. Continue to think. I'm going to continue on, but please continue to think. So looking to the future, we can carry this idea of compounding a step further. One application of pharmacogenomics is to be able to determine the optimum combination of drugs delivered at the optimum dose at the optimum time using the optimum release rates and so on, all based on the patient's genome and enzymatic and other information. Once these characteristics are known, a small high-tech vending-like machine, kind of like the 3D printer that I showed you this morning, printing a pill, can be used to manufacture, whatever the heck that means, enough customized pills for a given individual on an individual scale in the pharmacy or better in the doctor's office. Now think about this not so hypothetical scenario. You get sick, you go to the doctor writes a script, but unlike in the conventional model where we take that script to the pharmacy, he or she sends that information to a high-tech vending-like machine, i.e. a 3D printer, in the back of his or her office. Those particular pills are manufactured exactly for that particular patient, the number of pills they need, the number of different drugs they need, the number of release rates, and so on and so on. Another thing that's driven me nuts for a long time in pharma is why the heck should a pharma company decide how much medicine to put in a particular pill? This makes absolutely no sense. The doctor should do this, not the company. Oh, come on. Everybody's different. Okay, see, now I'm just posing the idea. Yeah, please. Neither do I. Okay, well, so again, I understand your point. I'm not going to mention a statistic, 99% or whatever, but I will respond this way. And remember, I teach medical school part-time as well. I'm constantly reminded of the old joke, what do you call the person that graduates last in their class in medical school? Doctor. I'll say it again. What do you call the person that graduates, with all due respect, what do you call the person that graduates last in their class in medical school? Doctor. You can say the same thing. I'm sorry? No, you're missing my point. I'll say it a third time and then I'm going to continue on. What do you call the person that graduates last, the bottom of their class in medical school? Doctor. But we can say the same thing about attorneys, about lawyers, and dare I say it, about regulatory professionals as well. This is another thing that drives me nuts. A lot of companies will say, we got FDA approval, we got FDA clearance, we're ISO blah, blah, blah certified. That is the academic equivalent of being a C student. That is passing. That's all that it is. That's passing. Something to think about. That's right. That's right. As I say to my medical students, every surgeon has graduated from medical school, but does that mean that every surgeon is equally as good? Of course not. There are a lot of regulatory professionals, a lot of regulatory consultants out there, but are they all equally as good? Something to think about. So how will we regulate this? What would be the regulatory burden? In other words, if we require companies to follow the traditional drug model with extensive clinical trials, I guarantee this technology, as I just described, is never going to see the light of day because it doesn't make any sense. Or are we going to regulate it as compounding has been regulated in the past, what I call sort of quasi-regulated for centuries? Does it matter if pills are manufactured by some company in some far-off manufacturing location, or by a high-tech vending-like machine in a doctor's office, or by hand by the individual pharmacist? Are they just not iterations on the same theme? And by the way, these are not just issues that we as an industry need to decide and to argue, but these are issues of healthcare policy that we as a country or perhaps as a world needs to decide as well. So hold on. So if high-tech compounding is not manufacturing, can or should FDA even regulate it? And if they don't, could this lead to another New England Compounding Center tragedy? Please, Miriam. Yep. So to be fair, Miriam, I'm trying to, as we approach the end of the day here, I'm trying to get us out of our little regulatory hypothetical universe that we live in and think about the real world and focus on what really matters most. To anybody outside of these walls, if I asked John Q. Public or Mary Jane Smith on the street if there was a difference between these things, what do you think they would say? I'll leave that as a rhetorical question. That's the point that I'm trying to make. And if regulation doesn't make sense, do we want to continue to perpetuate it? Absolutely. Absolutely, and I agree with you 100%. And, believe me, as I said this morning, I'm trying to make the world a better place. It is not an easy thing to do. I'll be a real quick, real story. I will be the first to follow the regulation when it makes sense and when it's to my advantage. But if the regulation doesn't make sense, I will be the first working with FDA or Health Canada, whoever it is, to try to figure out a way that does make sense. More often than not, I'm successful. Occasionally, here's what happens. We were presenting a product. This was outside the U.S. I won't mention which country. It doesn't matter. We said, with all due respect, your regulation does not make sense, and here's what we think does make sense in the alternative. The folks on the other side of the table, they said, we agree with you, Mike. And by the way, rarely do people even say that we agree with you, that the regulation doesn't make sense. But here's what they then said. This is in the regulation, so you've got to do it anyway. At that point, with all due respect, I knew I was dealing with nobody more than a two-bit bureaucrat. Somebody who knew that as long as they followed the rules, they weren't going to get fired. Never mind if, people died. As long as they followed the rules, they're not going to get fired. So what I said to the company was this. I said, look, we won. They admitted that they agreed with us. But it's their house, it's their rules, right? If you want to do business in that part of the world, you have to jump through this particular hoop. However, I also said to the company, you can try to make the world a better place. And this is, in fact, what they're doing now. Because what I suggested that they do is they get together with their competitors so that they can go to their particular government in this country. And you say, you know what, it's not just us that's saying this. It's these 4 companies together that say we represent 85% of the market share. This regulation doesn't make sense, and can we have a discussion as to how to make it better? But in the U.S., it's actually a little more complicated because oftentimes I hear people, get mad at FDA because, FDA makes them do something. More often than not, the beef is not with the FDA because FDA cannot do anything that Congress doesn't tell them to do. So oftentimes your beef is with Congress. And again, as I said earlier, if you think it's difficult to deal with FDA, which it certainly can be, that's nothing compared to Congress. Here's an interesting statistic for you. The pharmaceutical industry has more registered lobbyists in Washington, D.C. than the total number of senators and representatives combined. Now, that's not a good thing or a bad thing, but it is a fact. There's a lot of momentum wanting to continue the status quo. So we always got to look for precedent beyond medicine. We always got to look for similarities where they don't exist. So let's look for some sources of inspiration. Some of you, perhaps, if you're from the U.S., you may have seen this ad for Arby's on the TV.
My name is Bo Deedle. I'm a retired New York detective, and I'm slicing up the truth about freshness. A lot of people think the meat in this sandwich is sliced up in the back of this store. Let me show you where the back really is. This is where they slice the meat. In a factory. That's a long walk for a turkey sandwich.
Arby's slices fresh in store, not in a factory.
Slicing it in the store is exactly analogous to what I just described, producing or manufacturing, if you will, the drug in the pharmacy or in the doctor's office. So what's the message? How does it apply here? We've been doing these things in medical technology, not just for drugs, but for devices for a long time. But does it make sense? Not to me. There's got to be a better way. We always got to look for precedent everywhere. How many of you are familiar with the Coca-Cola Freestyle Jet machine? Watch this.
It was designed by the people who brought you the Ferrari. Gene Farrell is project leader. The heart of Coke's business has been falling in North America.
We hear from consumers all the time that the flavors, the combinations, and so on and so on. Watch
I haven't tried a regular Coke in years, but they've gotten me introduced to the brand because of this technology. And what we're seeing in our early testing is double-digit increases in sales for our customers.
The key is printer-style cartridges that release the ingredients in potent microdoses using technology borrowed from precision medical equipment.
So here we have cartridges of different flavors, cherry, vanilla, and so on and so on. Well, take out those cartridges of flavors and put in other cartridges of drugs. Lipitor, Prozac, Zoloft, and on and on and on. The idea is exactly the same. This is not nearly as far off as a lot of people think. Yeah? Well, again, let me be clear. Okay. Well, that's the point that I'm trying to make, is that take out Coca-Cola and put in Prozac. Oftentimes, there was a drug-eluting stent company called Connor that was subsequently bought by J&J by Cordis. They had what I thought was one of the most innovative delivery systems for their drug across the board, very different than most other approaches. Unfortunately, the drug they chose didn't really work. Therefore, the company went out of business. Imagine if you were bringing a syringe onto the market today. Syringes have been around for 100 years. If you were bringing a syringe onto the market and you put into it a particular drug and that drug didn't work, are you going to throw the syringe away because the drug inside of it didn't work? It happens a lot. That's probably what you're describing here. So let me just remind you, I'm not trying to convince you of the advantages and disadvantages of any of this. I'm simply trying to get you to think a little differently. So what else can we deliver in a vending machine? A lot of people say, well, drugs are pretty easy to deal with because they're very stable. I mean, you have to hit a drug very, very hard to get it to change structure. Biologics, on the other hand, anybody familiar with TGN 1412? I don't have time to get into that case. Biologics, especially things like monoclonal antibodies, inherently unstable. A small change in the temperature and the pH, as one of my friends at FDA likes to say, if you look at some monoclonal antibodies the wrong way, you could cause a change in the shape and the conformation of the protein. So how could we possibly deliver a biologic in something like a vending machine? Well, once again, I look well beyond the medical universe for inspiration. And when I do that, watch this. In this particular case, we're going to see, I think in Japan, delivering... I'm going to turn their narration off because I don't speak Japanese or whatever that is. But we're going to deliver live... I'm sorry? Shanghai. Okay, thank you. Okay, China, I'm sorry, my ignorance, I apologize. Anyway, they're going to deliver live crabs in a vending machine. You don't think so? Watch this. You got the vending machine here, you got these crabs inside there, they don't look like they're alive. You put your money in, you take out the crab, and watch what happens when they take the crab out of the machine. Just watch this. Just in a couple of seconds. The crab comes down. And I'm not showing this to gross anybody out. I'm really not. A live crab in a vending machine. I would argue that if we can deliver a live crab in a vending machine, we ought to be able to deliver any biologic that we want, including something very, very funky like TGN 1412. The question is regulation advancing? So the ultimate goal of pharmacogenomics, in terms of the practice of medicine, we want to deliver the correct drug at the correct dose, at the correct rate, at the correct patient, at the correct time. Very easy to say on a PowerPoint slide, by the way. Not so easy to do in the real world. And from a product development perspective, if we're going to fail, we want to fail early. We want to fail safely, that is, without killing too many people, and we want to fail without spending a lot of money. So bottom line, those are the ultimate goals here. The regulatory challenges, and I'm going to wrap this up, are profound. The FDA will need to turn the clinical trial paradigm on its head. This is according to Janet Woodcock, the top drug regulator at FDA. She said 2 years ago, we're going to have to change the way drugs are developed, period. This is the top drug person at FDA saying this. The FDA and drug developers will need a new definition of a clinical trial. Imagine the top drug person at FDA saying that we need a new definition of a clinical trial. The way trials were developed in the past should not influence or govern the way things are done in the future. For decades, FDA has requested 2 randomized controlled non-inferiority trials, but those may not be possible with the small number of patients possible in trials for personalized medicine. Remember, the epitome of personalized medicine, one product for one patient. What does the clinical trial even look like? This is going to require a grand change in the way clinical trials are thought about. It's going to be a big challenge for everybody, not just FDA, but for industry as well. My response, it's about time. Some of us have been singing these songs for a very long time, and it's about time that some other folks are starting to join the chorus. FDA 2 years ago put out this very interesting document. I don't know if it's still available on their website. They might have taken it off, but if you want it, I'll send it to you. Paving the way for personalized medicine. The thing I like about this is because it was the first major report at FDA to show examples of personalized medicine, not just from the drug side of the world, but from the medical device side of the world. Some of the examples that I've talked about this afternoon as well as this morning, and several of the examples that they talk about in this report, I personally had my fingers in. So this is happening, but it's taking a long time. Could things be finally changing at FDA? Peggy Hamburg, the former FDA commissioner, announced the formation of a program alignment group. She said this is a critical endeavor since more of the products that FDA is tasked with regulating do not fit into traditional categories, what I call silos, that FDA has historically divided its work. Devices, drugs, biologics, and so on and so on. Many new medical products transcend the boundaries between drugs, devices, and biologics. Many cosmetics, this is why I asked the question earlier, many cosmetics contain particles that mirror the nanotech particles found in medical devices. Many foods have drug-like properties. Again, Hippocrates said, let your medicine be your food and your food be your medicine. In such a world, the boundaries between FDA's different centers may no longer make sense. Regulation is increasingly being based on the function of a product rather than the traditional definition, food, device, drug, and so on and so on. But will FDA have the fortitude to break up its existing functional divisions and adopt a real program orientation? It's unlikely to happen anytime in the near future. By the way, this is not a challenge just for FDA. In some ways, this is a bigger challenge for industry. Another thing that drives me nuts is why do so many people continue to think that you work either in the pharmaceutical industry or the biotech industry or the medical device industry? In the future, we're not going to have any such thing as a medical device company or a drug company or a biotech company. We're simply going to have what I call medical technology companies that have components of all three of those things. And by the way, when I talk about the future, I'm not talking about 100 or even 50 years from now. I'm talking about even as we speak. And I could share with you a litany of examples, but I'll share with you just one. One of the companies that I've been working with for several years was started by 3 people, a mechanical engineer, a pharmacologist, and a molecular biologist. Now, what the heck is this company? Is it a medical device company, a drug company, a biotech company, all of the above, none of the above? I have no idea. But that is clearly the future. There's going to be a lot of internal resistance. A major reorganization will create a lot of dislocation. Large bureaucracies, whether we're talking about a government bureaucracy like FDA or a big drug or a medical device company, it doesn't matter, doesn't respond well to that sort of dramatic change. That is an interesting challenge. And if this happens, and unfortunately it's making, if the program alignment group was in a race with a snail, the snail would win. But at least we're making progress. Okay, so I know it's very, very late, but I'm sure you've had enough. Let me just end with a couple of quick final thoughts. As I said earlier, there are a lot of regulatory consultants out there, but there are not that many good ones. As I said earlier, every surgeon has graduated from medical school, but does that mean that every surgeon is equally as good? No. So how do you become a good one, or how do you find a good one if you need one? Very simple. Learn when to follow, but more importantly, learn when to lead. As I said earlier, if it's to my advantage to follow in somebody else's footsteps, then I'll be the first when I go into FDA's door to put in 72-point font in my PowerPoint. I'm doing nothing more than what was done before. Don't let the door hit you in the you-know-what on the way out. But if it's not to my advantage, or if it's not possible to do what was done before, I'll be the first to propose an alternative and to work with the agency to make that happen. Because if you want to lead the orchestra, you must first turn your back on the crowd. Something to think about. If you want to lead the orchestra, you must first turn your back on the crowd. And finally, don't just follow the rules, think. Douglas MacArthur said rules are mostly made to be broken and are too often for the lazy to hide behind. You can imagine the response that I get from this slide when I show it at very large conferences like RAPS. People throw things at me because they think that this is heresy. But I genuinely believe this. If the rules make sense, then by all means follow them. If they don't make sense, working with the agency, whether it's FDA or Health Canada or whoever, we need to figure out another way to make it happen. Again, we're well over time, but I'm happy to stick around for a couple of minutes, or if anybody wants to get a drink at the pub. I will be back tomorrow to do a workshop that's going to be interactive with some case studies. I'm going to ask you to come up with some regulatory strategies to bring some interesting products onto the market. But if anybody wants to stick around and ask questions, please feel free. Anybody? And thank you for those of you that did ask questions already. Yes, sir.